Alrighty then, we are going to shake things up and put glucose to rest for a little bit and shift the focus of our discussion to a new class of biomolecules, namely the lipids. Today's lesson is very heavy on definitions, and that's because there's many types of lipids and they do many different things. The class of biomolecule may be different now, but the molecule at the moment is perhaps familiar. It is lauric acid, and that is everyone's favorite fat compound derived from coconuts. And we're going to talk about its fuel potential right now. Complete oxidation of this 12-carbon fatty acid is going to give us theoretically 78 ATP molecules. Later on, we're going to talk about how we get that number, but for now, just take that at face value. And what we just learned in cellular respiration, that glucose, a 6-carbon compound, is going to yield about 32 ATP molecules max, which is considerably less than what lauric acid has to offer. So if you normalize the amount of ATPs for every carbon in the molecule, we're getting about 6.5 ATPs per carbon in the case of our fatty acid. However, glucose is only giving us 5.3 ATPs per carbon. My question for you is, why does lauric acid provide more energy per carbon than glucose does? Chew on that, and we'll discuss it in class. Now, what is a lipid? Most of us hear lipids and we think fats, and sure, that's one specific kind, but there are several lipids out there. And in general, my definition of what a lipid is an amphiphilic or hydrophobic biomolecule with a large ratio of carbon and hydrogen to oxygen. It's hard to get more specific than that because the types and functions of lipids are really quite diverse. We'll start by outlining what fatty acids are, and then waxes and triglycerides, and the whole way down this list, all the way down to the terpenoids and steroids. And as we take this tour across nature's many lipids, we will take note of which functions are associated with each of these classes. And you can see that the functionalities are quite diverse, ranging from, yes, energy storage, but also insulation, water repulsion, compartmentalization, signaling recognition, pigments, chemical attractants, antioxidants, vitamins, and this list is not even exhaustive. Your objective by the end of this class will be to look at a lipid's molecular structure and be able to classify what group it falls in and what roles it can undertake. Fatty acids are the simplest type of lipid and the building block to many others, so that's where we're going to begin. These are amphiphilic long-chain carboxylic acid compounds, and the emphasis is on long. Their hydrocarbon tails are going to contain anywhere between 4 to 36 carbon units long. And, that's not a hard cutoff, but that's a commonly reported range. And narrower than that, most of them fall between the C12 to C24 range. And you may notice that a lot of them are even numbered. I want you to put that in the back of your mind for now. That's not a coincidence. But anyway, here's a table of some common fatty acids that you will encounter in this course and just general biochemistry. Lauric acid, we just discussed, that's a C12. The technical IUPAC name is N-dodecanoic acid. I certainly don't expect you to remember the trivial names, which is really just where they were first isolated from, or the IUPAC names for that fact. Just be generally aware that this is how we name them. Not only that, fatty acids, that hydrocarbon tail, it can be either saturated or unsaturated, meaning we can have double bonds usually in the tail portion. So let's look at this fatty acid. It's got 18 carbons in it, all right? And we have a double bond, specifically a cis double bond on the ninth carbon extending to the 10th. So we can name this cis-9 octadecanoic acid. Now, I'm not super concerned with your ability to whip out IUPAC names at the drop of a hat. And that's in part because biochemists came up with I think, a more convenient notation system for describing the geometry and composition of an unsaturated fatty acid. And the rule is X colon Y parentheses delta Z. What the hell does that mean? X is simply the number of carbons. Y is the number of double bonds. And Z is the position of the double bonds along the main chain. So let's apply that to this compound, which I happen to know is oleic acid, by the way, abundant in olives. But the shorthand nomenclature for that fatty acid is either oleic acid, cis-9 octadecanoic acid, or 18 1 delta 9, which I think is much easier. Whether or not that alkene is cis or trans is going to impact its physical properties as well as its biological ones. Let's run with oleic acid again as an example. It's an 18 carbon fatty acid derived from olives, and it's going to have a cis double bond in the ninth position. What does that cis double bond do? It's going to impart a kink to the molecular structure of the hydrocarbon tail. And as such, you can envision that the bent nature of the hydrocarbon tail is going to preclude its ability to sort of stack up and maximize those van der Waals contacts, making it a more difficult compound to crystallize into a solid. This is manifested in the melting point of oleic acid, which is 13 degrees Celsius. That is less than room temperature, and hence why cis fatty acids tend to be oils or liquids. Now, let's compare this to elytic acid, its isomer. This is also a C18 fatty acid, same molecular formula. It still has one double bond at the ninth position, except in the case of elytic acid, it's trans. How does this impact the molecule's geometry? Well, you can see that you can draw it in a linear fashion way more easily. You could probably even imagine that it's going to stack up in this laminate way quite readily, like brickwork. We can more easily envision how these molecules will come together and crystallize, and therefore its melting point is going to be higher, 45 degrees Celsius, north of room temperature. So fats like these are going to be solids. Trans fats have more in common with saturated fats than cis fatty acids. I want you to now ask yourselves, comparing trans fatty acids to cis fatty acids, which of these two do you think is more likely to clog your pipes? Well, it's going to be the trans fatty acids. These are the less healthy ones. They are going to raise your bad cholesterol, raise your triglycerides, and are pro-inflammatory. We make trans fatty acids through the food manufacturing process. Cis fatty acids are naturally derived. We convert fatty acids into either saturated or trans fatty acids during food processing, ultimately for convenience. Solid fats are going to spoil more slowly than liquid fats, and they are less messy to handle and easier to weigh out and portion. So we take the hit in health because ultimately they're they're cheaper materials to utilize and they last longer, and I guess they improve the texture of the food as well, so on and so forth. Whether or not that's worth it is a separate debate altogether. Omega fatty acids is another common nutritional buzzword, and these refer to highly unsaturated fatty acids that feature many alkenes spaced at every third carbon from the very last or omega position in the hydrocarbon chain. So if we look at alpha linoleic acid shown below, that's the omega position. 1, 2, 3. Oh, look, there's a cis alkene. 1, 2, 3. Another cis alkene. 1, 2, 3. The third cis alkene. Why are omega or polyunsaturated fatty acids so important? Well, it's because animals actually struggle to make these. We often lack the enzymes necessary to do so. And we need these polyunsaturated omega fatty acids to make many essential hormones or neuronal membrane components. That's why we say they're good for your brain, right? So the only way to actually acquire them and incorporate them into our bodies is through our diets. Therefore, they're considered to be essential. Plants and algae are usually really good at making these omega fatty acids. Fish is only a good source because they eat a lot of algae, certain kinds of fish at least, and therefore they will be rich in these omega fatty acids by proxy. You might be disappointed to learn that free fatty acids are not that common in biology. They're usually kept stored up in more complicated fats like waxes and triglycerides. That's really where you find fatty acids in nature. But when it's time to digest, let's say, a triglyceride for food, we can crack them off and use them as substrates for a process known as beta oxidation. We will devote an entire lecture to beta oxidation. It's basically the process by which we respire fatty acids and convert them into chemical energy or usable chemical energy, ATP. However, we do occasionally see free fatty acids obtain a functional role, namely as signalers. Free fatty acids, such as arachidonic acid, can get released and participate in some key physiological pathways, particularly relevant to the cardiovascular system. And we'll talk more about these in a later slide, but they are going to be very relevant in vasoconstriction, vasodilation, and also inflammation. So these are very medically relevant compounds as well, and not just in a having too many fats is bad for your cardiovascular system kind of a way. I'm going to circle back to arachidonic acid eventually. In the interim, I wanted to discuss the next level of lipid derivative, and that would be the waxes. Waxes are incredibly hydrophobic esters of fatty acids condensed with fatty alcohols. And a fatty alcohol is just a fatty acid that has been reduced. So what are some examples of that? Fatty acid like palmitic acid is shown over here. That is a C16 fatty acid. And 1-triacontanol is an example of a fatty alcohol that's got 30 carbons in it. So altogether, triacontanol palmitate is a C46 wax, which has quite a bit of hydrocarbon content to it. And as a result, there's going to be a lot of van der Waals contacts between individual molecules of a wax, which is going to result in a very firm and stiff kind of an architecture. And that's useful for bees, for example, because this wax is the primary component of beeswax. It's what they use to build their hives. Not only is it stiff and strong building material, it's going to be very hydrophobic to the point where it's actually water repelling. That's also of benefit because you wouldn't want your house to wash away every time it rained, right? And the benefits of waxes can be found outside of hive insects like bees and wasps and such. For example, preen oil is secreted by birds to keep their feathers water repellent as well. There are many benefits to that. If the feathers get sponged up with water weight, that's more bird that it's going to have to fly and lift off the ground with their wings. Not only that, water is a decent conductor of thermal energy, so by coating their bodies in this preen oil, they won't get wetted by the water they swim in, which will help keep them warm and dry. In 2010, when British Petroleum accidentally spilled 200 million gallons of oil into the Gulf of Mexico, it dissolved off this protective wax, this preen oil, on, the birds that were in the Gulf. And those birds that weren't scrubbed clean of that wax died of hypothermia. We also see waxes in the plants, for example. If you ever washed leaves of a salad or lettuce, spinach, you may notice that the water beads on the leaf like so. And that's because they're often coated in a waxy material. And there's a benefit to that as well, and it has to do with mitigating water weight. Cellulose is polar. It's going to soak up water like a sponge. And you don't want that because every time it rains, that's going to make the leaf very heavy and bogged down with extra water weight, which can cause the leaf to snap and break. There goes your photosynthesis. If you cover the tops with a layer of wax, the water just rolls right off and the leaf won't get damaged.
The next class of lipids on the docket are the triglycerides. And these are what most people probably think of when they hear the word fats or lipids. They're also known as triacylglycerols. And that's because we form them by taking three acyl groups from fatty acids and condensing them onto a common glycerol backbone. Glycerol, shown over here in red, is a very special triol. It's got three hydroxyl groups, shown over there. And each of those alcohols can condense and esterify with a fatty acid that's going to result in this E-shaped molecule. And whenever you see an E-shaped molecule, that's usually a sign that what you're dealing with is a triglyceride. In terms of molecular composition, they can be simple, where we have one glycerol esterified to three of the same fatty acid, or they can be mixed, where two of them are the same fatty acid or all three are different. Perhaps you got a mix of unsaturated and saturated, so on and so forth. This over here is an example of a mixed triacylglycerol. It contains stearic acid, linoleic acid, and palmitic acid. Now, a quick disclaimer, do not let the presence of these three ester groups deceive you into thinking that triglycerides are polar compounds. They are not. They are very, very hydrophobic. Remember that those esters are buried beneath quite a bit of hydrocarbon content, and they're also vastly outnumbered. So therefore, biology has to store triglycerides in often a separate and dedicated tissue because these compounds are just not going to mix with our aqueous selves. In the case of animals, we drop off triglycerides into adipose tissue, which is shown over here. It looks like these white bubbles, and those bubbles aren't air or empty space. Those are actually globules packed full of triglycerides. Now, in the case of plants, they don't have adipose tissue, but instead they usually reroute any synthesized lipids into the seeds, for example, seeds and nuts, because those are going to require lots of energy to fuel germination once they're planted. So that's why peanuts, walnuts, sunflower seeds, they're all rich in triglycerides and calories, for example. Now, back to our functionalities of triglycerides. So the main function of these triglycerides are going to be energy storage, and particularly we're thinking long-term energy storage. Glycogen, it's constantly being made and broken down, made and broken down. Adipose is something that we're going to tap into not as a last resort, but as like a second resort once the glycogen runs out, basically. And at this point, I'd like to discuss the relative costs and benefits of storing chemical energy in the form of, let's say, glycogen versus a triglyceride. So, the perks of storing energy in glycogen molecules is accessibility. So glycogen equals water soluble and therefore accessible. Thanks to their abundance of heteroatoms, they are going to be very polar, very reactive, and so enzymes are going to be able to make short work of these polymers and be able to break them down and liquidate the glycogen bank. The drawback is that storing energy in glycogen is, however, inefficient. And that's because we have partially oxidized our food molecules, which lowers the amount of energy they could potentially possess. That's the price we pay for making them reactive and soluble in water. Now, in terms of triglycerides, these are not water soluble and less accessible as a result. I mean, take a look at this triacylglycerol. There is not a lot of polar atoms present activating our molecule. So the enzymes have a hard time reaching it. And once they do, it's like, what do they got to work with? It's a bunch of carbon-carbon, carbon-hydrogen bonds. Very boring, very stable. But there are perks to having a very inert food storage molecule. Yes, the energy density of this food storage molecule is efficient. because these are way more reduced than glycogen is. But not only that, the extreme hydrophobicity of the triglyceride molecule means that adipose tissue doesn't carry a lot of water weight. Low water weight. I mean, think about it. The lack of abundant hydroxyl groups means that triglyceride globules can't harbor extensively hydrogen-bound solvation shells around them, not in the way that glycogen does. So that means when animals store calories as adipose tissue, it's less water weight they need to schlep around while they hunt or run from predators. There are evolutionary benefits to not having this extra weight. So that about sums it up for the ways that animals versus plants store triglycerides. We also can't forget about triglycerides' roles in insulation and buoyancy. In the winter months, humans do tend to pack on pounds more easily, not just because of holiday feasts, but as an evolutionary relic. We have evolved to protect ourselves during the colder months, and one strategy for doing that is through insulation. And true, we don't hibernate still, and we don't use thermogenin or brown adipose tissue to warm ourselves without shivering, but there's still benefits to having the extra padding in the colder months. Now, animals that live chronically in Arctic conditions, like this really cute baby seal. I love when they have the three black dots, is what I call it. They're so cute. They spend most of their lives in frigid water, and if they're not in frigid water, they're on top of a frigid iceberg. So they need to be insulated from all that cold, and that's why they tend to be very plump with this adipose. And thermal insulation isn't the only benefit. Oil is less dense than water, so being rich in adipose tissue is also going to make the mammal buoyant. These are not fish, seals, whales, etc. They need to come up for air every now and then, and fat tissue makes it less strenuous to get back to the surface for another breath.
Now, phospholipids, which is a subtype of membrane lipids, are structurally related to triacylglycerols, except they're actually a diglyceride composed of two fatty acids and a phosphate group occupying that third slot in the glycerol backbone, which is going to give this molecule a charged and polar region and a very hydrophobic uncharged region. And therefore, it's going to be amphiphilic. And that amphiphilicity is really important for enabling its role as a membrane lipid, and for two different reasons. Number one, we've all seen diagrams of phospholipid bilayers by now. They look kind of like this. We've got the charged head groups facing the water on the outside, and then the hydrophobic tails all pointing to the interior. And then we get that same arrangement again on the other face. And so what we're doing here by positioning the polar head groups on the outside and the tails on the inside, we're dissolving the exterior of the membrane in both the cytoplasm and the extracellular matrix, which are going to be aqueous. So the polar head region is going to partially solubilize the membrane, and we need it partially soluble. The other benefit to having that polar head group is it's very tunable. We can swap out what I've shown here as an X with a number of organic entities, which is going to give us different types of phospholipids with different types of functionalities. And they can do many cool and interesting things, such as form dedicated compartments within the endomembrane system, and maybe even serve as signaling molecules. Let's talk briefly about one of my personal favorite phospholipids, cardiolipin. This is a really dramatic example of a crazy looking head group. Its head group is technically a conjoined phospholipid, where the phosphate group of one diglyceride is going to be conjoined to another. So that's going to result in this cone-like geometry of the fatty acid called cardiolipin. And you can kind of envision how if you align these together, that's going to induce sort of a curvature in the geometry of the lipid bilayer membrane that they compose. And that is what enforces the cristae of the inner membrane of the mitochondria, which allows us to have lots of surface area and maximize our electron transfer chain potential. So something as simple as the geometry of the fatty acid can install improved functionality inside our own organelles. Now let's talk again a little bit about the signaling capability of these phospholipids. I told you that cardiolipin is something that's found in abundance in the inner membrane of the mitochondria. Let's say that mitochondria is experiencing a lot of redox stress, lots of radicals. There's actually mechanisms in place that can send cardiolipin to the outer membrane of the mitochondria. And the cell knows that cardiolipin belongs in the inner membrane, not the outer membrane. And if the cell sees cardiolipin on the outer membrane, it can actually induce apoptosis, controlled cell death. That could be a sign that the cell's becoming cancerous and we're doing aerobic respiration faster than we should. Let's shut things down before the problem becomes a catastrophe. Sphingolipids are another subtype. The membrane lipids. And they have a lot in common with the phospholipids. The main structural difference is that these are actually derivatives of ceramide instead of phosphatidic acids. They don't have a glycerol backbone, but rather a sphingosine backbone, which I've shown here in red. And most noticeable is the fact that we're conjugated already to this unsaturated fatty acid, and our second OH group has been replaced with an amine. Now, it's a little confusing because these two categories aren't clearly cut apart. Like sphingomyelin is actually both a sphingolipid and a phospholipid because its head group is phosphocholine, which takes the place of this X placeholder over there. But not all sphingolipids have phosphates in them. That's really more of an exceptional case. Functionally, sphingolipids are major constituents of membrane lipids and cellular markers in particular. GM2 ganglioside is a negatively charged glycosphingolipid that contains this sugar called sialic acid, also sometimes called neurominic acid. Bacteria don't have these sphingolipids, so vertebrates use this sialic acid to tell their immune systems that we're not pathogens. Anything without this sialic acid or this GM2 ganglioside gets attacked by the immune system. Cancer, however, is conniving, and it has a tendency to overproduce or upregulates the production of sialylated gangliosides, and that helps it evade our immune system. And not only that, there's a negative charge imparted onto the GM2 ganglioside, seen over here. It's a sugar acid, and that's going to induce a degree of charge-charge repulsion between individual cancer cells, which is going to weaken cellular adhesion and thus promote metastasis.
All right, we have a lot going on this slide. Next up is the eicosanoids, and I want you to recall arachidonic acid from a few slides back. It's a C20 fatty acid and also the precursor to four diverse groups of lipid signalers that all contain 20 carbons in them as well, hence why they're called the eicosanoids. Eicosan is, I believe, Greek or Latin for 20. And all of these eicosanoids are going to be formed by a class of enzymes known as cyclooxygenases. We call them COXes for short. And what COXes do is oxidize or epoxidize arachidonic acid in many different ways, which results in four types of eicosanoids. So let's first just define what they are and what their general anatomies are. We'll begin with prostaglandins. These are very identifiable because they possess five-membered rings in their head groups, shown over there. Very similar to the prostaglandins in form and function are the thromboxanes. These contain, however, six-membered rings. And also an epoxide, very common within them. Third up are the leukotrienes. As the name suggests, they consist of three alkenes conjugated together within the arachidonic acid backbone. And the last class are the lipoxygens. These are going to be polyhydroxylated arachidonic acid derivatives. Many OH groups are decorating that hydrophobic tail of the eicosanoid. So now let's take a moment to walk you through all the different kinds of functionalities these eicosanoids can perform. And we could dedicate a whole course or two just to this class of molecule alone. But we're going to keep it simple and just do the surface level basics here. So let's start off with the prostaglandins. These are notorious for being pro-inflammatory signalers. And how do they achieve that effect? Well, through smooth muscle contractions. So what do smooth muscle contractions do to you? Well, it depends on where you experience them. If they happen outside of your cranium, that's going to give you a headache. If you are on your period, smooth muscle contractions are what causes your uterus to shed the unimplanted lining that you developed throughout your cycle, and it will also make you experience painful uterine cramps as well, unfortunately. Those are both good examples of localized smooth muscle contractions. What about experiencing them systemically? That's going to cause your whole body to shake and shiver, which is going to increase your body temperature and result in a fever. Fevers are bad for you, but hopefully they're going to be way worse for whatever pathogen is infecting you. And lastly, these prostaglandins are going to make you feel like crap. It's going to make you want to stop exerting yourself, stay in bed, focus on recovery, and be extra sensitive to pain. We call that hyperalgesia. So it's meant to help you, and it does. It's a necessary evil, but some of us don't like to suffer. So what we do is self-medicate with an over-the-counter class of drug called non-steroidal anti-inflammatory drugs, NSAIDs for short. These are medicines like aspirin, ibuprofen, Aleve. And how these operate is they are suicide inhibitors of the types of COXes that convert arachidonate into prostaglandins. So it's going to reduce your fever, make you hurt a little bit less, maybe ease your headache, so on and so forth. Moving on to the thromboxanes, what these are famous for is localized vasoconstriction. You know when you get a paper cut and you're like, oh, maybe it's not going to bleed, and then seconds later it does? We call that hemostasis. And what's happening there is the injury is going to release thromboxanes, which is going to cause smooth muscles lining your broken capillaries and such to contract. And that's going to dam the flow of blood for a couple of seconds. What that achieves is it gives your body time for the clotting factors to ramp up and get a head start on clot formation. This is why, by the way, that blood thinning and bleeding is a side effect of NSAIDs. These are very promiscuous COX inhibitors, and they're going to dampen your pain, but also attenuate your ability to clot. Thromboxanes are also able to elevate blood pressure through a similar mechanism by spasming the smooth muscles around capillaries and blood vessels. You're going to reduce the volume, which is going to increase the pressure of the contents inside. Another example of a good vasoconstrictor are the leukotrienes. These are a little bit different though, because leukotrienes tend to act upon the respiratory system more than our extremities. If you have seasonal allergies, allergic rhinitis, the puffing up of your sinuses and bronchi, you have leukotrienes to blame for that. If you have a really severe sensitivity to environmental allergens, those people can end up with asthma, an impaired ability to breathe because of all of this swelling that's occurring in our respiratory tissues as a response to the vasoconstriction. So it can lead to bronchial obstruction. And in super severe reactions, it does also play a role in anaphylaxis. So what's a common way of treating these unfortunate symptoms? Well, we don't necessarily, to my knowledge at least, use COX inhibitors, but we can use montelukast, aka Singulair, which competes for leukotrienes at the receptors it triggers, and it prevents it from turning on that allergy switch. And it's right in the name, montelukast, leukotriene. That's not a coincidence. So you can think montelukast, leukotriene, allergies to help you keep it straight. Now, lastly, the lipoxins are the black sheep. Their functions are various. They're generally not potent, not as well understood, not as well easily classified, but we have seen that they do have roles in vasodilation and anti-inflammatory
inflammation. And this is interesting. NSAIDs, specifically aspirin, actually activates lipoxin generation. That's why low-dose aspirin, baby aspirin, is often prescribed to people with cardiovascular disease. It's going to open up their blood vessels, reduce blood pressure, while dampening the ability to form clots, while decreasing the inflammation that is scarring or affecting the arteries of that patient. Cardiovascular disease is an inflammation by nature, and so that is a very simple solution to a complex problem, or at least part of a solution to one. So yeah, the eicosanoids, they're very important to medicine, which is why they are incredibly druggable targets. Them and the cyclooxygenases that make them. Terpenoids, also known as terpenes, are really cool molecules, in my opinion. Unlike everything else discussed in this lecture so far, these lipids are not made from fatty acids. Instead, they are derived from this hydrocarbon shown here called isoprene. Isoprene is going to have two reactive terminal alkenes that are able to link up with other isoprene units via addition reactions, and the ensuing products are often very volatile, very hydrophobic, and have many different uses. Let's consider, for starters, small isoprene derivatives, which are the components to many essential oils that you may know and love. Flavor compounds in our spices, perfume compounds in our colognes and cosmetic products. Citral is a compound that gives lemongrass its flavor and scent. And it is essentially derived by stitching together two isoprene units. Very simple modifications to one terpene changes its molecular structure and therefore changes which olfactory sensors get triggered in our noses. By basically adding a bond here and then turning this into a hydroxyl, you convert the lemongrass scent, citral, into the pine scent, alpha terpenol. And then another modification to that small terpene derivative turns pine scent into camphor, the scent of Vicks VapoRub. I don't expect you to know the complicated mechanisms by which these molecules interchange, but I do expect you just to be vaguely aware that a lot of these compounds smell amazing and therefore attract pollinators to the plants to help them spread their pollen. Or it provides a nice flavor that incentivizes animals to grow, harvest, and disseminate these plants. Now, what happens if you chain up a bunch of isoprene units in a row and make a very long polyalkene? Well, because these have higher molecular weights, they're not going to be as volatile, but by conjugating a bunch of alkenes together, they're going to now absorb very intensely within the visible spectrum of light. And that will ultimately give you pigment compounds. We all know that beta carotene gives carrots their orange color, but it also makes the petals of the poppy plant colored, and it will color many other fruits. And again, the pretty colors are going to attract pollinators and animals to pick up the fruits, transfer the seeds,
and help that organism survive across multiple generations. But remember, of course, the goal of many plants is to get animals to eat and spread the fruit and seeds, not the entire plant. You don't want it eaten root and stem. That's where elastomers and repellents, such as polyisoprene, aka latex, comes from. Perhaps the pollen and flowers of the latex tree smell and look nice, but we don't want animals eating the useful parts of the plant, like the bark and stem and roots. So the latex tree is going to create this polymer of isoprene and inject it into the sap. So when animals try to bite the tree and get a bunch of sap in their mouth, it's going to glue their mouths shut, and then they die. And so it trains local fauna to eat this part of the plant, not that part of the plant, otherwise suffer the consequences. And the ironic thing is, us humans, being inventors, realized that we could use this very springy, elastic sap for our own purposes. And therefore, we grow latex trees in plantations and harvest this sap that was initially meant to be a deterrent for animals. Plants aren't the only organisms that use terpenes, not by a long shot. Many of our fat-soluble vitamins, for example, are terpene-derived. Remember our friend ubiquinone, coenzyme Q, from the electron transfer chain? Well, it consists of a redox-active head group that is attached to a very hydrophobic polyisoprenyl tail. Look at that. The hydrophobic character of the isoprene units shown there is what ultimately helps dissolve and anchor that portion of the molecule to the inside of the inner mitochondrial membrane. And that's what ultimately allows electrons to be ferried between complex 3 and complexes 1 and 2. And even beyond that, more generally speaking, isoprenyl tails are sometimes attached to proteins that are meant to be directed to the surface of a membrane in general. So this is a very useful strategy that biology has learned to utilize. What other vitamins contain terpenes? Vitamin K, which is used to facilitate the action of thrombin, the enzyme that basically finalizes the blood clotting cascade. That also contains a polyisoprene-derived tail unit. Vitamin E is an antioxidant. It also has a polyisoprene component. And here's vitamin A, retinol. It's essentially a beta-carotene molecule that's been chopped in half. And this is used in the vision transduction cycle of certain proteins in our eyes. So check it out. The functions of terpenes isn't just to look good and smell good. They help us do respiration. They help protect our bloodstreams by facilitating clotting. They protect us from oxidative stress. And they help us see. Pretty nifty. The last type of lipid on the docket are steroids. These are actually derivatives of a special terpenoid called squalene, which is basically 6 isoprene units that are linked together. We are going to talk more about steroid synthesis in a few lectures, but I want to just briefly introduce you to it here to get that ball rolling. If you take a polyisoprene molecule like squalene and you wind it up in this zigzag manner, you're able to take an enzyme and zoop, form a bunch of bonds like so, and get a very complicated steroid nucleus out of the reaction, which ultimately gets matured in many steps into cholesterol, which I like to call the mother of all steroids. And a steroid is basically any molecule that has these four few rings, A, B, C, and D. Usually there's some additional action over there on the D ring, and very commonly you will see a hydroxyl or a carbonyl on the A ring. And the functions of these molecules are so diverse. I mean, cholesterol does a bunch of things, like regulating membrane fluidity, for example, but the molecules that the body cholesterol gets converted into are responsible for signaling, hormones, vitamin precursors. The list goes on and on and on. Cholesterol often gets a bad rap, but it's actually a crucial component to mammalian physiology. To prove to you just how useful and modular the steroid backbone is, I'm going to show you four very similar looking steroid hormones that have very different functions. Let's first consider testosterone and estradiol. These hormones are responsible for sexual maturation and development of secondary sex characteristics often associated with these genders. We also have aldosterone, which has nothing to do with the reproductive system. It regulates salt retention in the kidneys. And cortisol, which is a decent anti-inflammatory, and it regulates glucose metabolism and chronic stress response and a bunch of other things. Structurally, all of these compounds are quite similar, but they all have vastly different physiological effects, and they're able to foster that because of just slight modifications to our backbone. Cholesterol derivatives can also have non-hormonal uses, and these are going to be the basis for most bile salts. Bile salts help with digestion by emulsifying the fats that we eat. We'll talk more about that later, and they're able to do this thanks to their highly amphiphilic nature. Cholic acid is one type of bile salt, and what we do here is oxidize the alkyl tail attached to the D ring and imparts an explicit charge to that side of the molecule, making everything else hydrophobic and that part hydrophilic. It's basically a good biological soap, if you will. And there's several other cholesterol derivatives out there that I don't have time to even list, and you're not expected to know them, or the structures of these derivatives, or their minutiae. All I want you to know is what the general structure looks like, and that their uses are multifarious. I always like to end on a practical note or zoom out into the big picture. And so you're likely already aware that hyperlipidemia, hypercholesterolemia are both big problems in the industrial world. Bad genetics and or a bad diet causes cholesterol to accumulate in our bodies, and that puts us at risk for heart disease. Cholesterol alone kills 4.4 Americans every year. And that statistic came from the American Heart Association back in 2019. That number has likely increased since. Yet I believe it was the early 2000s when scientists discovered that plant cholesterols called stanol esters were a thing and they were pretty unique to plant physiology. They're not utilized by the mammalian body. In fact, stanols, an example of one is shown over here, they compete for the cholesterol uptake receptors lining our guts. So, some entrepreneurial scientists decided to incorporate stanols into butter alternatives like Benecol, shown here. And you can buy this in the supermarket right now. It's a little pricey, but I actually like how spreadable it is. And no, I was not paid to give this endorsement or advertise for this company. I just think it's kind of cool that practical biochemistry can be found on the shelves of your local supermarket. By spreading this butter on your toast every morning for a month, you are going to be inhibiting competitively the cholesterol uptake receptors in your intestines, which has been documented to reduce your systemic cholesterol levels by 10%. And you can see the results published here by, a reputable group in 2003. And I don't know, I think that's kind of interesting. It's also just such a comically American invention, for us to find a way to make a type of fat that you could eat that reduces the amount of fat in your bodies. It's really, though, a clever twist to have a cholesterol reducing butter on the market. Anyway, I'm not saying to go out and buy this immediately. At the very least, I'd like you to maybe think fondly about biochemistry the next time you're strolling down the dairy aisle. Although, if you are a Benecol executive, I will end this by saying that I happily take both checks and cash.